Hello, everyone, and welcome to our third pitch session of the Invest in BC conference presented by Lumira Ventures. This session is going to take a little bit of a different format. It is supported by Global Affairs Canada, the Trade Commissioner Service, and we are going to have a mix of presentations as well as a couple of pitch sessions. So hope you enjoy this session. We will first hear remarks from Philip Brunswick, the Trade Commissioner of Life Sciences at the Consulate General of Canada in Boston, and then Jenny Karnajanovija, Trade Commissioner, Pacific Regional Office with Global Affairs Canada. From there, we'll welcome Dr. Qing Zhang, partner at LDV Partners, to provide a presentation of her own. And then she has personally selected 3 of our pitching companies to present within this session, where she'll conduct a live Q&A with them. Of course, for the investors in the audience, your interest in these companies is also always welcome. So please be sure to post any questions that you have in either the chat or the Q&A function found at the top right-hand corner of your screen. Without further delay, I'd like to welcome Phil and Jenny to the stage. So a little bit about their background as they turn on their cameras. Phil serves as the Life Sciences Trade Commissioner at the Consulate General of Canada in Boston. In this role, he actively works with and supports Canadian life sciences companies that are seeking to grow their business through partnering, capital raising, sales, and technology commercialization in New England and the USA. And Jenny is a trade commissioner based out of Global Affairs Canada's Pacific Regional Office. In her current role, Jenny is responsible for supporting Canadian life sciences companies to do business abroad. Jenny and Phil, I'm going to turn the virtual stage back over to you.
Great. Thank you so much, Wendy, for your introduction. Good afternoon, everybody. My name is Jenny Karnajanovija, as you know, and I'm a Trade Commissioner with Global Affairs Canada's Pacific Regional Office. Today's session is sponsored by the Trade Commissioner Service. As many of you already know, the Trade Commissioner Service works to support Canadian companies to expand their exports into markets abroad. We are a free service, and we help Canadian companies, much like yourselves in the room today, to do business in markets overseas. And we do this by connecting you to funding and support programs, as well as international business opportunities. And that's done through our network of trade commissioners in over 160 cities worldwide. In this session, we're very excited to have LDV Partners listening to pitches from 3 innovative Canadian companies. They are H2O Biosciences, Renaissance Bioscience, and Vesalius Cardiovascular. Before we begin, though, I'd like to extend a big thank you to Qing Zhang at LDV Partners, who has generously volunteered her time to be at our event. LDV Partners is a US-based VC firm that invests in early-stage companies across a range of sectors. Qing will be providing a presentation shortly, so I won't say anything more. But before that, I'd like to turn it over to my colleague, Phil Brunswick, who's been working with me on this event for his remarks. So over to you, Phil.
Great. Well, thanks, Jenny, and very nice to be here. A great program, and great to be connecting with the teams out in BC. And as Jenny mentioned, I am the Life Sciences Trade Commissioner based here in Boston, and I am one of many life sciences trade commissioners based in the US as well as globally. So within the United States, we've got roughly 16 missions across the country, most with life sciences trade commissioners. And as Jenny was mentioning, our primary objective is to facilitate trade, product development, financing, and market entry into the United States. So as a life sciences trade commissioner, I basically cross all boundaries of life sciences. So I do a lot of work on the biotech side, therapeutics, diagnostic, drug discovery as well. We just wrapped up a big conference here in Boston called the MedTech Conference, where we had roughly, over close to 200 Canadians come down to participate in this event. Unfortunately, not too many west of Ontario, but next year the MedTech Conference is out in Anaheim, so I expect strong BC participation in that event. And then the year following, it's actually in Canada, in Toronto. So big plans for the MedTech Conference. And I guess the one key area that I'd like to kind of leave with everybody is think of the Trade Commissioner Service, and particularly me here and my team in Boston, as a free and valuable support group for you. You know, we're here to help you and support you, and we do anything from helping with your funding, your financing, but more importantly, making connections. For me in my territory, which is the Northeast area, but as well, we collaborate regularly across the country and across the world. So we are here to support your activities. My background is all in life sciences. I only joined the consulate a little over a year ago. So for a number of years, we'll say prior to that, all in the pharmaceutical biotech side of the business in Canada, then transitioned over to the medtech side, worked for Boston Scientific for about 13 years, first in Toronto, then out to Minnesota, Minneapolis on the cardiovascular side. And that's kind of how I got here to Boston. And I've been here in Boston in the life sciences industry for the past 16 years. So think of us as experienced people who have the desire and capabilities to help you guys grow and develop and find your niche here into the United States.
Perfect. Thank you, Phil. For those of you who would love to learn more about the Boston Life Sciences ecosystem or about some of the events that Phil just mentioned, please be in touch and we're more than happy to help. It is now my pleasure to introduce you to Ching Jiang, partner at LDV Partners, and Ching will be providing an overview of her company. So Ching, please turn your camera and mic on and you have the floor.
Thank you so much, Phil. Much, Jenny. Hi everyone, this is Cheng. So nice and such a big honor to be participating in this great event. I would love to introduce you all to LDV Partners. We are based bi-coastal in the Bay Area and also here in Boston. And our thesis, actually the fund was named after Leonardo da Vinci, that we would love to invest in modern day Da Vinci's. Next page, please. Right, so it's a general fund, and we do early stage investment from ideation stage, like pre-seed and typically through Series A. We do participate in later rounds opportunistically if it's a good fit. And we mainly focus on two sectors, deep tech and also bioengineering. And we will categorize all these into 4 buckets. The first one is robotics automation across different industries. Second part, we love the data and how to monetize data. And that leads to something about Web3 and FinTech. And third part, bioengineering covers everything in life science and healthcare, from life science tools and services to diagnostics to medtech and new modality of therapeutics. And the last part, but not least, is advanced industrials. It's a combination of biotech and other specialties. Say, for example, food tech, agricultural and industrial tech as well. As a fund, we love the barrier of technology, and we believe that new technology can usher in new business models, can make disruptive change to the current best practices. And I'd love to highlight one fact that we are extremely founder friendly, and we believe that founder is at the core of innovation, and they're really the drivers behind. Next slide, please. So on this slide, just to give you a case of how we kind of like cut the value chain in different industries into particular sectors, and how we believe what kind of technology is the best fit into what bucket. Say, for example, in healthcare, throughout like the progress of disease from prevention, diagnostics, recovery, payment, and enabling, and the new therapy discoveries, there's a ton of new technologies that can be potentially commercialized and applied to help people and to save lives. Next slide, please. Is it all? Okay. So the last page, this is just a showcase of our portfolios across the many years of experience between my colleagues. So the two partners, the two founding partners, in particular, they're both deep expertise in physical sciences. And we have people background in biology and healthcare like myself, and also a couple more colleagues specialized in AI, machine learning, and engineering in general as well. Yep. So, and we also do like early stage working with academics to help them commercialize their patents to spin off into a commercial company. So that's why you see like the stealthy mode ABCD on the top. That is more of a heavy lifting, but it can be very, very rewarding in many ways. And we seek to continue doing that with academics and professors that we have the mutual trust with. Yep, so I wouldn't dive into the portfolio companies for now, and looking forward to learn more about what's going on in the market, and definitely can't wait to hear from the founders who have like exciting stories to share. Thank you very much.
Perfect. Thank you so much, Jing, for that excellent overview. And yeah, now we will begin the pitch process. So our first presenter of the day will be Anthony Fayez from H2O Biosciences. So I'd like to invite Anthony to come to the stage and deliver his presentation. So over to you, Anthony.
Thanks very much, Jenny. And thank you, everybody, for giving me the opportunity to participate in this session. So to begin with, my name is Anthony Fayez. I'm the CEO of H2O Biosciences. And of course, we are a deep tech science company. So our goal, of course, is to more accurately model and predict the behavior of atomic interactions, which is the core of molecular modeling and drug optimization. As a team, we're quite small. We were founded in 2020, and we've grown to 8 people so far. And we recently finished raising funds in April of this year. We raised $1.5 million US. So we are not currently seeking funds. However, that may be that status may change in 2023. Ultimately, our goal is to be able to do cheaper, faster, and better molecular modeling, which typically is unusual. Most groups are able to do 2 of the 3, while our goal is to do all 3. And currently, we're looking for strategic R&D collaborations to help enable rapid drug development. So let me delve in a little bit more. The problem that we're actually working on is for drug optimization, and nearly all the tools out there currently use the same equation. This is pretty much considered the traditional model for molecular modeling. We're not going to get into the math today. I don't want to delve into that. However, there really are only 5 terms in this equation describing the different components of interaction. And unfortunately, all 5 of them have known sources of error. They don't work the way that we expect them to. Thus, the traditional modeling packages that are pretty much used ubiquitously all have the equivalent, they all have this particular source of error baked into them. And our goal, of course, is to remove that. So I'd like to focus just a second on the computational chemistry ecosystem, because there really are 3 components to it. The first is what I just described, the traditional molecular modeling area. That's where they're using that equation. You apply it to understand the interactions. And of course, with the known error in it, it has only so much accuracy that you can get out of it. On the other end of that spectrum, QM, or quantum mechanics, allows you to predict the behavior of atomic molecule or atomic components, but it only scales so high. So you really are struck with the limit that you can't scale to large molecules, which is typical in biology problems. You just cannot apply QM to that. There are various groups who have tried to break out of that mold, but none so far has really managed to encapsulate the accuracy of quantum mechanics with the speed and scalability of molecular modeling. So our goal is really to do that. There's a third component here, which is AI. There's a tremendous number of AI companies out there, some of which are in our neighborhood. And we're very happy to work with them. We don't really see them as competitive. We see them more as complementary, because nothing we're doing stops them from doing what they're trying to do. And much of what they're trying to do actually works well with the technology that we're building. So really, the future where we're trying to take this technology is not just limited to doing better drug optimization, but to take it in directions that are currently impossible with the existing tools. A lot of molecules out there, or drug targets out there, are based on targets that have relatively deep clefts in them, which is where you'd stick a drug molecule in to gum up the work, so to speak. of how a druggable target would work. A lot of targets, however, or the vast majority of them, have very shallow or flat surfaces. And in order to access that and drug them, you'd have to really understand how the water works in that case. The solvation plays a much larger role, whereas when you have a cleft, the solvent, the water, is unable to get in there. So you can more or less rule it out, which is okay if you're using a traditional molecular modeling package. They don't really understand solvation particularly well, which leads you to the point that it's really difficult to make those models work well for the undruggable ones. So that's what we're aiming for in the long term. Just a couple of quick points on what we're different on. We have built what's called a force field. That's a different set of equations that governs the behavior of molecules. And we focused on three things. Ours have atomic charge fluctuations, breakable bonds, and the goal, of course, at the end of the day is to be able to convert, to produce emergent properties, so we can understand exactly how the pieces are working together, even if we don't have an accurate training set, which is not really required for molecular modeling, but would be for AI. So just focusing on that for a second, dynamic charges is really important, particularly for solvent work, and of course for when you're working in interfaces. And that's really where we want to go in the long term with the undruggable targets. So we've put a lot of effort into that, and we can demonstrate that our platform does work well in that situation. The other thing that's important is we believe in bond breaking, which is also not a part of traditional traditional molecular modeling. And for us, that plays a large part in micro pHs and how the interactions on surface chemistry really work. So we've put a lot of effort into that. And we do, in fact, have the ability to demonstrate we can work on this and correctly produce breakable bonds. And in terms of being able to produce emergent properties, we didn't train our molecules on water specifically, nor on ice, and yet we're able to take our molecular modeling platform and demonstrate the correct behavior of water at low temperatures and at high temperatures as well. So that works out really well, and we can demonstrate that water being, of course, key in molecular modeling and chemistry. If you can demonstrate how it behaves properly, you can really extrapolate and you can predict how the molecules should behave, whereas traditional platforms really don't do a great job at that. Just in order to compare ourselves with our competitors, we do have the ability to quantify the error that we have. We use the generalized Amber force field test, which is, of course, produced by their test, not one that we've created ourselves, and we're able to outscore both them as well as the Merck Frost force field on most of the tests that we've been using. So that gives us a great confidence that our platform is working. And of course, the goal at the end of the day is to match what quantum mechanics is able to predict. So this is just one example of torsional profiles that we can do. Not particularly important other than to say that we are in fact demonstrating that we can match what quantum mechanics would predict as we'd expect. The force field itself is completely novel. It's not based on an AI approach, though we do use machine learning in order to parameterize it. That allows us to work on some very interesting chemistry because we have very few atomic types. So compared to Amber, there's a lot of parameterization required. Our force field is much easier to set up and to work with, which is fantastic because it also extrapolates further than Amber does, which is exactly what we wanted. At the end of the day, we're looking to fit seamlessly into the drug discovery workflow of our partners. So given a specific target protein and some candidates, we should be able to do ranking, we should be able to do insight development, and that gives us the opportunity to iterate and ask new questions. So we're exactly fitting in with the existing workflow for our partners. And our core business model at the end of the day is twofold. We believe that in the beginning, we're going to be able to work on contract research where that insight is useful and the drug ranking provides a lot of value. However, in the longer term, as I mentioned earlier, we do want to build our own in-house drug development platform, which would allow us to go after the undruggable targets. Just a quick note on our timeline. We expect to have our MVP ready by the end of this year. We're currently in the beginning of an early adopter program, so we're looking for partners to help us validate the platform and get us through commercial launch and into our first couple of commercial projects. And again, somewhere in 2024, we expect to start working on our own internal drug development pipeline. So what we're looking for specifically, we're looking for early adopters and people who are interested in using our application. We're not selling our software. We're looking for people to partner with us. The software will stay in-house with us, but we can certainly work on projects for our partners. We're also looking for collaborations, of course. We want to be able to put together a long list of useful applications for our platform. And if there's something that you specifically think would work, please feel free to reach out. And of course, we're also looking for other positions as well. We're looking to fill a last board seat. We would like to grow our science advisory board. We're very likely to have open positions in 2023 to join our team. So thank you very much. Much appreciated, and looking forward to some questions and participating further.
Great. Thank you, Anthony. Ching, I'd like to invite you back to the stage for the Q&A process. So please feel free to, perfect, you're here. We have about 4 minutes for Q&A. So over to you, and feel free to ask any questions as you please.
Thank you so much, Anthony, for sharing a fascinating technology. I'm definitely a layman about quantum mechanics, but it's fascinating. We'd love to learn more. So maybe just very quickly on the question side, curious, like in your software and in your potential partners, what kind of like partners is ideal for you? Like those partners with like, are you working on potentially new target identification or best in or known target, but it's tough to drug or undruggable, and or are you looking to improve the current properties of current small molecules?
Right now, we're actually looking for a good validation project. So we're looking to start small. We know that there's a lot of skepticism that we can outperform everybody else. That, of course, is reasonable. So we're taking a measured approach. We're in the process of putting together white papers to demonstrate the technology works. Then we'll work on a validation project. And then we'll look for relatively conventional projects where we can actually contribute value. Once we've demonstrated the platform and we've continued to refine it over the next year or so, then I think we can start working towards the undruggable ones. But we have to set our sights realistically and make sure we hit all the steps in the right order. So that's what we're trying to do.
Right. So is it fair to assume that you can possibly start with some like drugs that failed in either phase 3 and then try to optimize it, and then I see. Yeah,
absolutely. There's a lot of applications for this, both in terms of drug repositioning or drug optimization. We're not doing ADMET stuff yet. That's further down the road, but hopefully one day. So in the meantime, we're going to be focusing on improving binding, improving scoring and ranking, and all these things fit really nicely into our model and the software we've developed.
Right. Yeah. Great. Is your so you only work with small molecules? Is that correct? You don't work on.
For now, it's a matter of scaling. There's no reason our technology won't work on protein-protein interactions as well. We can already demonstrate that we can manage small proteins, but the goal really is to start with small molecules, because I think that's the most tractable problem and the one where we can generate the most insight in the short term. And as our project scales and our platform scales, then I think we'll be able to get into much larger molecules.
Sure, and maybe even like RNA targeting small molecules as well, right? For that's an interesting space for sure. Yeah, definitely
on our list. It's already there.
Yeah. Last but not least, curious about your comment as the insider of the industry that with DeepMind, publishing their huge library, what is that gonna influence or impact our industry?
It's a huge, huge impact, actually. If you look back at drug development for the last, 40, 50 years, the computational side has been really slow to develop. And a lot of that's because we just don't have good structures for target molecules. Now we have access to pretty much everything out there. So this is just feeding in exactly where we are now. We want to be able to look at everything out there and pick the best targets. Now that we have structures, we can go after them and start drugging them as well.
Awesome. Thank you so much. No other questions? Any questions for me or?
Not right now. Thank you.
All right. Wonderful. Well, thank you so much. Oops. There we go. Sorry about that. Thank you so much, Ching and Anthony, for your questions and for the presentation, Anthony. We'll be moving on to the second presenter, Devana Welton, CFO of Renaissance Biosciences. So over to you, Devana, for your presentation.
Thank you for the introduction, Jenny. And thank you all for taking the time to listen to our presentation today. Renaissance is a life science company situated on the beautiful UBC campus. And we focus on the bioengineering of yeast to provide differentiated solutions and sustainable solutions to a wide range of billion-dollar market opportunities. Our company has been around for almost a decade with the same CSO and founding employees who understand the yeast genome and are expert in the field of whole organism engineering, genetic engineering, and combining the two. We're somewhat unique as we have already actually created and commercialized what we refer to as our Generation 1 technologies in the food and beverage space. And now we're using our experience gained from our research and development all the way through to commercialization to apply it to our Generation 2 technology, which is our patent pending yeast-based production and oral delivery system for RNA bioactive molecules. So in terms of our business model, we focus our efforts on where we have the expertise, and that's on the research and development of a technology platform that creates our yeast solutions for unmet needs in various industries. What we then do is partner with large global organizations who have the means and the experience to perform the other commercial product functions, such as the regulatory, the production, marketing, and sales. And then we monetize these agreements through a combination of success payments, milestone payments, and back-end royalties. And we find this business model is advantageous to us at our current stage of development, as it allows us to tackle numerous diversified targets from our proprietary platforms, and thereby we can increase our probability of success, and we can create multiple revenue streams. And it also allows us to have a lean cost structure as our industrial license partners manufacture and distribute the end products while we shift our focus to the next viable yeast solution. So using this model for our Generation 1 platforms, we've created patented and licensed yeast technologies for the food and beverage industries, which has resulted in about $4.5 million in revenue to date. So our beverage portfolio includes our hydrogen sulfide preventing yeast, in which we've created a number of strains of production, and we've licensed over a dozen of these strains. And interestingly, for the beer drinkers out there, in 2022, our beverage licensee launched our new lager strain, which is classified as a novel Group 3 strain. It's considered the first new commercial lager strain in over a century. And then our licensed acrylamide reducing yeast for the food industry is used in standard manufacturing processes by replacing traditional baker's yeast. And what that does is reduce the amount of the acrylamide found in our carbohydrate-rich foods. And acrylamide forms when certain foods are cooked above 120 degrees centigrade. So it becomes present in things like French fries, toast, and coffee. So this technology is really important, as acrylamide exposure is linked to cancer and it's toxic to the nervous and reproductive systems. And importantly, these food and beverage revenue streams are expected to increase over time with minimal effort from us. Our beverage revenue will grow as our superior strains continue to gain traction in the market, and we introduce 1 to 2 strains per year from our platform. And our acrylamide reducing yeast will increase as the market grows from tightening government regulations over this carcinogen. So what now? Everyone at Renaissance shares the same belief that it's possible to produce the things we need using biology in more efficient and sustainable ways without an undue cost on end users. McKinsey and Company refers to this as the biological revolution, and we have a huge opportunity to utilize our platform to contribute to the annual economic impact expected to be up to $4 trillion a year from the 400 estimated use cases, for which we're creating solutions for crop protection, animal health, and human health. And so how are we doing that? Well, by now, most of us have heard about power and also the historical difficulties of using RNA commercially. To date, it's been sparingly used in industry due to the fragile nature of RNA in its natural form, which results in a difficulty in creating an effective delivery mechanism into the target organism, as well as the cost of manufacturing a full delivery system. However, investments starting to ramp back up in this area due to the effectiveness of RNA, the increased public awareness of the benefits through the mRNA vaccines, and some of the successes that have been seen in the market. And at Renaissance, we have spent the last number of years creating a solution to overcome the obstacles associated with RNA applications, all while using our favorite simple baker's yeast. Our science team's been able to modify the yeast genome, which already harbors RNA naturally, by turning on or off different genes to heighten the amounts of RNA to commercially feasible levels, which is what our patent, which is pending, pertains to. We then add the target-specific RNA effectors to our yeast, and then that creates our complete bioengineered yeast system containing RNA effectors. And an important question is, why yeast? Well, because it's one of the most well-known, studied, and consumed microorganisms on the planet. It's food-grade safe, and it's found everywhere. It's in our food or wine or coffee fermentations or chocolate fermentations. And really importantly, yeast has a very robust cell wall, which allows us to protect the RNA in really harsh conditions, including inactivation, freezing, and boiling of the yeast. And this allows us to orally deliver RNA bioactive molecules like never before. And with the ability of yeast to be compatible with short and long RNA effectors, we're able to use our one production and delivery platform to create valuable strains for different industries. And also, as yeast is inexpensive to produce, as it's already done on a large scale globally from a byproduct of the sugar refining industry, we're able to manufacture high amounts of valuable RNA at a lower cost. So So our population is growing and is forecasted to continue to grow with an increase of 23% anticipated through to 2050. This significantly increases the burden on global agriculture as we don't have the ability to meaningfully increase the amount of arable land available to us. And in addition, regulations are tightening around the use of chemical pesticides, sometimes without there being a solution. So we basically have to find a way to protect our crops and increase yield without damaging our planet or its inhabitants. And so approach to addressing these crop losses in a sustainable way will be cost competitive while also remaining effective and increasing safety compared to current solutions. Because we're using RNAi to shut down a specific protein or number of proteins required for life, we're able to target and eliminate a specific crop pest without affecting another organism, such as beneficial insects and humans. And also because RNA is a nucleotide and is biodegradable, we'll also be able to virtually eliminate harmful residues left on the crops. And so this is just showing you a test that we did with our yeast-based RNA biopesticide technology, which we performed on Colorado potato beetle larvae, which provided a very impressive result. It was 98.3% mortality. It was more like 100, but there was some wiggling around. And we were able to significantly protect the potato leaves, which is the important piece. And then in terms of animal and human health, the current advances in RNA-based therapies have substantial promise in treating and preventing many diseases and disorders through fixing the path instead of merely just treating the symptoms like many conventional options. And this technology has potential for substantial benefits as it will be delivered orally, which is much less invasive. It has the potential to reduce off-target effects as it downregulates a specific protein, and we believe it can be done at a competitive cost when compared to current alternatives. And then just showing you another graph here. We've been able to show that our yeast-containing RNA has the ability to knock down both TNF-alpha and IL-1 beta, which are inflammatory cytokines that are considered responsible for Crohn's. So finally, what are we raising the money for? While sometimes in biology we need to let nature take its course, we would like to be able to accelerate as fast as we can. Our platform is ready, and with the additional funding, we'll be able to progress each of our pipeline targets more efficiently in tandem, including sample creation for potential partners and regulatory submissions, which will lead to material revenue generation at a faster pace. And this is integral to success as proof of concept results in each of these categories, whether it's biopesticides, animal, or human health, is mainly what will drive licensee interest and commitment. Myself and our experienced leadership team would just like to thank you for taking the time to listen to this presentation, and I hope to connect with some of you further after the conference, and I look forward to your questions.
Great, thank you so much, Devana, and just bringing Ching back in the stage for the Q&A. Over to you, Ching.
Thank you, Jenny. Thank you so much for sharing, Devana. It's a fascinating, very powerful platform that addresses agriculture, food, animal health, and human health, like all in one. That's super powerful. My question is mainly from two folds. The first part about the technology platform per se. You mentioned, seems like it's all DS, like double-stranded RNA. Wonder what is the pattern look? What does the pattern family look like? Is it like, gene editing and usually new plasmid and then turn out to generate those RNAs? And how do you actually modify the yeast, or how many species of yeast do you have in-house, it's proprietary? Yeah.
Yes, we have a ton of yeast strains in-house, and what they've done is, through adaptive evolution, is kind of found a way to kind of go in and modify that yeast genome so that it knocks down certain genes within the yeast that are basically using that double-strand RNA for the viruses. And I think that's what the patent pertains to, is those particular genes and knocking down those genes that's resulted in that increase of RNA, because all yeasts have that double-stranded RNA, but it's not commercial. So our patent is basically around those particular genes that we were able to identify and modify so that we were able to increase the levels of that.
Right. Great. Thanks. That's helpful. And the second part is more about the pipeline development, because among all the things you can do, why did you pick pesticides or biopesticides in a way, right?
Yeah. So when we were, we've always had this blue sky dream of we would like to work in human therapeutics. But when we were doing all of our testing and we're using insects as bioassays, we started looking at it saying we're very, very capable of creating really specific solutions that will go after bugs. And, with the UN's plan to try and eliminate hunger in a sustainable way, and, with that push for sustainable solutions that are needed in agriculture, we just felt that this is a great way for us to drive value and create revenues for us while we're working on the human side, which is a longer regulatory process. We've got to go through FDA. And so this helps us kind of prove out the platform and do the proof of concept to show people that, yes, we want to build something, we build it, and we were successful in animal health and then down the road, right?
Yeah, perfect. I totally agree. I think climate tech in general is such a important and huge topic, and government level, they're willing to invest and to support the industry to grow. So I believe this is a very good starting point that you picked, right? And in terms of animal health, what kind of products are you looking at?
So we've kind of got two on the go right now. One is aquaculture. So we're doing white spot syndrome virus in shrimp. So we've actually been able to show that our yeast containing RNA can go through their feed process. It can get, tossed in, go through the high friction, high heat, and still come out and be fed to the shrimp. And our RNA is protected within it. So that's one thing we're looking at, because there's currently no solution. That's over $1 billion industry there. And then the one thing we're actually looking at is working on porcelain epidemic diarrhea virus in pigs. So we're actually working with the Vaccine and Infectious Disease Organization out of Saskatoon, and it's a big item that back in 2013 there was over $1 billion of losses from numerous pigs that ended up suffering from this. So that's one thing that we're looking at as well.
Great. And then my last question is about commercialization in the market. Are you looking at Canada? Are you looking at US? Do you have a particular region that you're interested in?
Yeah, so the partners that we work with generally are big global companies. So, it might be that a particular pest might only be in one area, it might only impact Europe or something like that. But we would like to go after the, really big companies that have the global reach. And so when they're distributing their products, they have the ability to get big market share. So we would like to be global. And we do have some of our partners, we have one in Canada, but we do have a couple internationally already for our Gen 1 technology.
Yep. Great. Sorry. Well, actually, one last question regarding the food sect. I don't know if you have plans to continue working on food, but say if you do, is it considered as a GMO, gene modified, or no?
So we started off with our acrylamide reducing yeast was GMO, but now we have non-GMO patent on that as well. And we will do it opportunistically if it's high value to us, right? Like we have that capabilities. And so we wouldn't, risk kind of shareholder money to do that. But if someone's coming and offering us a joint development agreement and something that we're very experienced in, then we will do it. And, we do, like we say, we do have our beverage strains that we'll just release 1 to 2 strains per year as our license. Literally costs us nothing. So yeah, we'll definitely do those opportunistically.
Sounds like a good plan to bring in revenue streams. Yeah.
Mm-hmm.
Awesome. Thank you so much for sharing.
Yeah, thanks. Appreciate getting to do the questions with you.
Thank you. Yep. Back to you, Jenny.
Perfect. Thank you. Thank you so much, Devana and Ching, for that. I'd like to bring our last presenter to the stage, Vincent Ledoux, COO of Vesalius Cardiovascular. So hi, Vincent, and over to you for your presentation.
Hi, Jenny. Thanks so much. Thank you again. So Vesalius is developing the next generation repair solution for degenerative mitral regurgitation. What is DMR? DMR is a valve leak inside our heart, and it's affecting millions of people around the world. Anatomically, it's caused by ruptured cords. You can see it on the slide. This disease is degenerative, meaning it doesn't stop progressing. It's very common. 5% of the population suffer from it, and it's fatal. The gold standard treatment today is mitral valve repair, and this is in open heart surgery. Surgery is effective, but comes with risks. Therefore, more than 50% of patients cannot undergo a surgery today. They cannot be treated. This is why DMR is currently heavily undertreated with an enormous unmet clinical need. If we explore further the gold standard treatment, open heart surgery, it is safe and effective thanks to two proven techniques: chordal replacement, so replacing the ruptured cords, and annuloplasty, which means reducing the size of the hole, the annulus. On the other hand, open heart surgery is risky because of the invasive access to the heart. It's a very big operation, as you know. And also it's risky because we stop the heart and we have to bypass the heart in order to repair the valve. Therefore, the perfect treatment is to provide the two proven techniques, so chordal replacement and annuloplasty, but with a catheter while the heart is beating. And this is the Vesalius mitral valve repair device. It has an annular ring, which provides the annuloplasty, a flexible and dynamic net, allowing a one-size-fits-all device. This is very important for inventory purposes. Multi-segment neocords or new cords, treating any disease complexity. So this is the chordal replacement part. This device is delivered to the heart with a catheter through the femoral vein. Basically, with one device, one size fits all, we can treat patients at any stage of the disease. This device that you see on the screen is at preclinical stage today and has been tested successfully in vivo 30 days, 90 days, even 300 days for some components. The test showed no injury to the valve, no thrombus, and very favorable histopathology results. If I want to summarize this in one sentence, our device is the first made-to-measure and leaflet-independent device providing comprehensive transfemoral repair for DMR. And this is a partial animation of our device delivery system. It's a 29 French system. After positioning the anchors inside the heart, we rail our device over guide wires, and our device deploys inside the heart with no risk of migration or movement. Following this first step, we lock our device. Today, you'll only see one lock, but we have several locks on the annulus. And we do the exact same thing on the lower part of the heart, the ventricle. We position the device where it should be. So this is a catheter-based task. Same thing is done on the other side of the ventricle. And since this device is pre-adjusted based on data from the patient, taken from the patient, it repairs the valve. And then if the patient needs annuloplasty, in the majority of cases, they need a reduction of the annulus, our device can provide annuloplasty again with a catheter. The procedure that you saw on the screen, or partial procedure, is exactly the same for each patient. The competition is very limited in this space because this disease and the dynamic anatomy are very complex. We have a first generation of transfemoral repair implants at commercialization stage, but because of the disease complexity, they have limited effectiveness by concept. Also, they are better suited for FMR, a different disease. A second generation of devices using proven surgical techniques, so chordal replacement and annuloplasty. This second generation is progressing around the first in a human stage. Vesalius is part of this group with one major difference. Vesalius has a significant technical lead by design. Our solution is the only solution that is leaflet independent, meaning we do not need to touch, interfere, or even anchor to a diseased, very fragile, extremely sensitive leaflet. And this is why we're the only solution that's leaflet independent today. We provide a comprehensive repair. Comprehensive meaning we reproduce the surgery. So both techniques used in surgery, Vesalius has embedded them inside of its device, while other companies are only focused on chordal replacement. Our device can treat any disease complexity. We have to remember that this disease is degenerative and progresses with time. We can treat the disease at any stage, while the other solutions can only treat one area that has been affected by the disease. And finally, we have a standardized procedure for all patients. So we have the unique solution that can provide surgical repair, like surgery, but with a catheter. Vesalius is currently a few months from first in human stage. We are planning our OUS, so outside US, study in Q3 2023, so 12 months from now. This will most probably be in Canada, 1 to 3 patients, one site. In parallel, our interaction with the FDA started 2 years ago, and we are progressing in our regulatory testing based on the agency's feedbacks. We are targeting an early feasibility study IDE in the US in Q1 2024 with 5 to 15 patients in 2 different sites. We are currently raising $20 million to finance these 2 stages. Vesalius, in summary, we have built a highly differentiated and elegant device respecting proven surgical principles and deliverables with a catheter. We have 5 major inventions in the mitral space that can potentially be used in other cardiovascular areas. We filed 18 patent applications. 3 of them has been already granted. And we performed dozens and dozens of in vivo studies to prove concept, durability, functionality of our device, including procedural and several chronic, 30 days, 90 days, and 300 days. And finally, we have a clear clinical plan. As a final slide, I would like to share with you that our solution has been selected and presented during the major European and North American cardiovascular events in 2021 and 2022. These events are highly selective and heavily scrutinized by physicians from around the world. Thank you very much.
Thank you very much, Vincent, and just bringing Ching back to the stage for Q&A. Over to you, Ching.
Thank you, Jenny. It is indeed very elegant and beautiful product, Vincent. Actually, let's take one step back. Would you mind sharing with us? I'm sure the audience will be curious to know the story behind this company. Like, you know, how? I mean, where is the idea? The invention coming from, even?
It's thank you for Ching for this opportunity and for asking this fantastic question, because I was saying what type of question Ching can ask me, and that was the first one on my list. This is why I was laughing. Vesalius started from the heart of the OR. In this field, particular field, cardiovascular, and the mitral space, mitral valve is the name of the valve. The domain experts are not only cardiac surgeons, they need to be specialized cardiac surgeons. They need to specialize in this valve specifically. And the co-founder of this company, Peter Skarsgård, is the veteran cardiac surgeon. And this is how the company started. It started from an observation, a discovery in the OR, where operation after operation after operation, Peter discovered a way to be able to reproduce what he is doing on a daily basis with patients, but embedded in a device that can be delivered with a catheter. And this is how it all started. And after that, some research were done, patent filed, of course. In this field, patent filing is in parallel to the R&D, and we started hiring engineers in 2017. So this is year 5 of engineering for the Vesalius cardiovascular.
Yep, beautiful. And regarding the progress of the current product, you have done first in human already in Canada, is that right?
Pardon me?
Have you done first in human trial yet?
Not yet. So the first in human will be in Canada in 12 months from now. We are preparing for it.
I see. Perfect. And actually, for the indication, just curious. I mean, what kind of patients would you enroll in? And is there any restrictions on the heart function, the EF, I mean LVEF and stuff?
A great question again. So basically, the first target will be patients who are not surgical. So this is the goal of this device, is to be able to treat patients who cannot undergo a surgery. And then after the clinical trial, we will be able to move to patients who can undergo a surgery. But since the surgery comes with a huge burden, the cost of the surgery, the recovery of the patients, about 3 to 6 months, the hospitalization, 10 days of hospitalization, et cetera, et cetera. Because of the burden, we will be able to move to non-surgical patients or surgical patients. After the non-surgical patient, we'd go to surgical patients. And even we will be able to treat patients early on in the stage of the disease. And this is extremely important. This disease is degenerative. The earlier is treated and the better it is for the patient. So this is the global target for our clinical study and the goal of this device.
Yep, and I assume it is regardless of underlying disease, whether it can be degenerative, functional or
there is one
related.
There is one not disease but bacteria where we cannot intervene, and even an operation cannot be done. And this is in between 1% and 3% of the patients where this bacteria is affecting the valve itself, and you cannot touch the valve. And this is the only major counterindication. The other counterindications are general ones, but the major one related to the mitral space is this specific bacteria that can affect the valve, and in this case, we cannot touch the valve.
Right. Maybe the only option is the surgery then, right?
Yeah. Even with surgery, they have to treat the bacteria before and eliminate it before doing the surgery.
So that's a complicated case. I love the fact that it's universal. It's not customized for all the patients, so that makes the doctor's lives way easier. It will be an easier go to market and ramp up. Indeed, yeah. And have you identified? Are you doing the manufacture in house, or are you working with CDMOs? And if you have you identified anyone to work with?
So for now, we are still in-house. So we have 8 biomedical engineers working on the device. Since the quantities that we need are for testing purposes and first in human, so they are still limited. So everything is done in-house for now.
Great. Yeah, we'd love to be in touch. Thank you for sharing.
Thank you. Thank you.
Back to you, Jenny.
Great. Thank you so much, Ching, and thank you so much, Phil, for the presentation. This brings us to the end of our session, and I would like to thank everybody for participating in our event. We've heard pitches from 3 very innovative companies, and I know that this certainly won't be the last time that we hear from these firms. To those of you that pitched today, thank you very much for telling us about yourselves, and Phil and I will be in touch shortly regarding next steps. Before I hand it over to Wendy for her final closing remarks, I'd like to extend a really big thank you again to LDV Partners, to Life Sciences BC, and to Phil Brunswick at the Canadian Consulate in Boston, because without their help, this event would not be possible. So thank you very much again, everybody, for attending and for listening. And I will turn it over to you, Wendy, for your closing remarks.
Thanks very much, Jenny. And thank you all for participating. Phil and Ching, your participation was really fantastic. Jenny, thank you for leading this session. And to Global Affairs Canada for allowing it to come together. It was a really interesting session. And to our presenting companies, Anthony, Devana, and Vincent, your pitches were fantastic. I hope that it was a great session for you, and having that opportunity of having the Q&A was really valuable. It was great to hear your answers, and so a big thank you to Chin for leading that. We now have dedicated networking time, and we invite all of the attendees to join our one-on-one networking session by clicking on the networking tab on the left-hand side of your screen, and you enter the session from there. You'll be randomly paired with another attendee for 5 minutes, and once that time is up, you'll be shuffled and paired with another attendee. We have found and that these sessions are really valuable, and continue to hear from people that they've had really interesting and valuable connections through that sessions. So please take advantage of it while you're on this virtual platform. You can also visit our info desk in the main lobby if you have any questions or need any more information. So I look forward to connecting with some of you during the networking session, and otherwise we will be back at 3 o'clock PST, which is an hour from now, for our final session of the day. Thanks everyone, and thanks again to Global Affairs Canada.